I'm Nicole Tessier, and in this video we will be looking at a brief overview of fat absorption. So in particular, we will be looking at what fats are, bile salts and how they are used in fat absorption, the enzymes involved in fat digestion, how fats are absorbed into the small intestinal cells or enterocytes, and finally how fats are absorbed into the body. And this slide just shows a nice image with the steps of fat absorption in a nutshell. So what are fats? Fats, or also known as lipids, are biomolecules made mostly of carbon and hydrogen atoms. They are nonpolar, which makes them not very soluble in water. And in the Western diet, fats are predominantly found in the form of triglycerides, cholesterol, phospholipids, long-chain fatty acids, and fat-soluble vitamins. In this video, we will predominantly be looking at triglycerides, as they are the primary form of lipids found in plants and animals. And there's just an image of a triglyceride on this page. You can see the glyceride portion of the molecule to the right, as well as the fatty acid tails that are extending towards the left. In this case, it is a saturated triglyceride, as the carbons in the fatty acid tail are predominantly single bonded. So fat digestion begins with chyme leaving the stomach, and it contains large, coarse fat droplets. And these fat droplets have less surface area than smaller fat droplets, and they are also water insoluble. Because of this, the liver will secrete bile salts, which are bile acids conjugated with amino acids. And these bile salts will break down the large fat droplets into smaller, more stable particles. And the result of this is that there is more surface area, and the fat droplets are now water-soluble due to the coating of bile salts that are surrounding them. Now, bile salts are amphipathic, and this means that they have a hydrophilic or polar region, as well as a hydrophobic or non-polar region. And when they form the coating around the fat droplets, as you can see in this slide, the hydrophobic portion interacts with the lipids themselves, while the hydrophilic portion interacts with the fluid surrounding the fat droplets. Now, the main enzymes involved in fat digestion are colipase and lipase. So the bile salt coating surrounding the fat droplets prevents enzymes from actually working on the fats. Because of this, colipase will displace some of the bile salts found on the surface of that fat droplet, and this will free up the fats for lipase to then act on those fats. And when they do that, lipase will remove 2 fatty acids from each triglyceride molecule, which will result in a monoglyceride molecule seen at the top right of this image, and 2 free fatty acids seen at the bottom of the right of the image. Now, the fatty acids will coalesce with the bile salts, mono and diglycerides, phospholipids, and cholesterol in order to form micelles. And micelles are droplets of phospholipids arranged so that the fatty acid tails are on the inside of the micelles. The micelles will then migrate to the brush border along the apical side of the membrane of the enterocytes, and there they will break apart, allowing the lipids on the inside to migrate into the cells themselves. Now, the lipophilic fats, which are the fatty acids and the monoglycerides, will enter the cell using simple diffusion. Once inside the cell, the monoglycerides and fatty acids will move to the smooth endoplasmic reticulum, and there they will reform the triglycerides that was their original form when they entered the body. Cholesterol, on the other hand, must enter the cell using active transport via energy-dependent membrane transporters such as NPC1L1, which is a transporter that can be inhibited by the drug ezetimibe, which will lower cholesterol levels in the body. And finally, there's the fat absorption into the body. Inside the enterocytes, the reformed triglycerides will coalesce with the cholesterol and other proteins to form large fat droplets called chylomicrons. And these chylomicrons are too large to be excreted on their own from the cell into the interstitial fluid. So instead, they are packaged into vesicles by the Golgi apparatus, where they will then be secreted into the interstitial fluid by exocytosis. Their size also prevents them from crossing the basement membrane of capillaries to enter the bloodstream. So because of this, they will enter the lacteal, which are the lymph vessels of the villi, and from there they will travel through the lymphatic system and enter the venous blood just before it flows into the right side of the heart at the vena cava. So in summary of fat absorption in the body, fats are emulsified by bile salts, which allows for an increase in surface area of the fats, as well as makes them water soluble. Colipase and lipase will break down triglycerides into one monoglyceride and two fatty acids, which will then coalesce with other lipids to form micelles. Micelles will move these fats to the brush border of the enterocytes, where the fats will then exit the micelles and enter the cells of the small intestine. Fats will then get packaged into chylomicrons and exit the cell on the basolateral side through exocytosis within vesicles. The chylomicrons from that will then move into the lymphatic system and finally into the blood.